I'd like to share with you today our results about the cellular landscape of Alzheimer's disease and disease-associated astrocytes. So my lab is positioned in Jerusalem at the Hebrew University, and we're focused on understanding age-associated neurodegenerative diseases such as Alzheimer's disease. So as you all know, we live in an aging world, meaning that the percent of aging individuals in the population keeps increasing and is projected to increase even much more over the next 30 years. So alongside this increase in the number of individuals also comes an increase in aging-related brain pathologies. And unfortunately, for most of these age-related pathologies, we currently don't have any treatments, any effective treatments or prevention strategies. So what we do in the lab is we try to tackle these interesting diseases by looking not at a single risk factor, but rather trying to look at multiple risk factors and their effect on the brain. So as you all can probably understand, the major risk factor for neurodegenerative diseases such as Alzheimer's disease, and specifically sporadic Alzheimer's, is actually advanced age itself. So aging is followed by molecular changes in the brain, which is really a huge risk factor for neurodegenerative diseases like Alzheimer's and other types of dementia, which we don't fully understand the link. But on top of the age, we also have many other factors that are risk factors. So we have a strong genetic component that is associated with the risk for Alzheimer's. And we also have other factors such as environmental factors and also lifestyle like diet and physical activity. So how to integrate all of this together is a very complex question. And what we'll do today is I'll actually demonstrate part of our work where we're trying to tackle Alzheimer's disease and really try to understand this disease in a very deep cellular and molecular level. So Alzheimer's disease is a progressive neurodegenerative disease of the old age, and it's the most common form of dementia. And it's associated with very distinct pathological hallmarks. So traditionally, what we believe is that the cognitive decline results from degeneration and damage to synapses and neurons, and later depth of these neurons that leads to the cognitive decline. And all of this is strongly associated to pathological hallmarks, specifically amyloid beta plaques and neurofilament tau tangles that are intracellular protein aggregates. But for over 30 years, the research has mainly been centered around understanding the effects of these pathological hallmarks and also the very neuronal centric. And what we know is currently we still have no effective treatments and prevention strategies, yet the rate in which people are accumulating this disease is very high, which is really kind of a call for action for us to try and look at the disease in a different way. And this is what we're doing in the lab. We're transitioning from a neuronal-centric approach to studying Alzheimer's and all of the other related pathologies into a more holistic approach where we're trying to profile the entire cellular environment. So if we look under the microscope into the brain, we wouldn't exactly see this cartoon image, but what we will see is that neurons are not alone in the cellular environment of the brain, but rather they're surrounded with multiple other cell types, including oligodendrocytes, that astrocytes, microglia, the immune cells of the brain, vascular cells, and others. And this cellular environment is critical for the healthy function of the brain. And the interaction between the cells are also critical for that. So it's very clear that if there's a damage to a specific cell type, such as neurons, the entire cellular environment will actually react to that. But what we are suggesting is that this reaction is not just kind of collateral damage, but rather it actually occurs early and it is part of the active cellular cascade that's leading to cognitive decline. And there's actually a lot of evidence already about the involvement of different cell types in the progression of Alzheimer's disease. And but we still don't have a very clear view of what is the cellular cascade that's leading to Alzheimer's disease. And our strategy in order to address this very big question is to track cellular and molecular changes in the entire cellular environment across different stages of the disease and along the process of aging, trying to find these molecular triggers and the key cellular components that are taking an active role in the progression of the disease. And the way we profile the entire cellular environment is using the single cell RNA sequencing approach, which enables us to really identify the cellular and molecular drivers. So one word about single cell genomics for those who are less familiar with it. So single cell, generally, when we try to find molecular differences, it's very common to measure molecules such as RNA that are highly informative about the state of the cells and processes that are going on in the cells. But when we're doing bulk RNA sequencing or bulk genomics, we're taking a piece of tissue and extracting all the molecules from this piece of tissue at the same time. So it's later very hard to relate a specific molecular change to the cell type where it arises from. For example, if we have a specific cell type that is changing, we might be able to track these changes in the bulk RNA sequencing, but we won't be able to say if this is a microglia cell that is changing, the neuronal cell, astrocyte, and so on. So what we want to do is want to actually measure these molecules, and specifically RNA molecules, in individual cells. And recent advancement in single cell genomics have really been enabling us to do this using microfluidic devices to capture tens of thousands of cells at the same time and measure thousands of RNA molecules for each individual cell. In the brain, we have an additional complication because of how hard it is to take brain tissue and dissociate it to single cells by using an enzymatic digestion. So what we've been developing over the year is a method called single nucleus RNA sequencing, where we extract the cellular nuclei, and similar to single cell RNA sequencing, we can profile thousands of RNA molecules from individual nuclei of cells. And we still get very similar information about which cells did we capture and what are they doing. And we'll actually see this. So just to complete the picture, once we've performed the single nuclei RNA sequencing approach, we then have a huge database of thousands of RNA across hundreds of thousands of cells. So we then need to apply machine learning algorithms on big data set in order to really extract the information. Today, I'll actually skip most of the algorithmic components, but they're always in the background. So what do we actually, just to summarize, what we're actually doing is we're using single nuclear RNA sequencing approaches to to profile the brain, to profile the cellular environment of the brain, and comparing disease to healthy brains in order to find cell types and molecules that are changing during disease progression. And actually, previous work has been recently published using similar such approaches, either nuclei or single cell RNA sequencing on mouse and human brains, have already revealed that multiple cell types are indeed implicated in the disease progression because we're seeing dramatic changes in them. So two such new, relatively new players that are really now at the front of the Alzheimer research are, first of all, microglia cells, the immune cells of the brain that have clearly been shown by work of other groups that they are acquiring a new cell state that's specific to the disease in Alzheimer's. And also oligodendrocyte cells are now turning out to also be incredibly interesting in their role in Alzheimer's disease. So we don't have the full story yet, but we're definitely getting towards it. And what I'll show you today is a new cell type also involved in Alzheimer's disease, which are astrocyte cells. So what we did in our research is we started in using mouse models. We took wild-type mice in an Alzheimer mouse model called 5XFAD, and we compared the cellular environment in the hippocampus brain region. So this is a collaboration between my lab and the lab of Dr. Michal Schwartz from the Weizmann Institute, Aviv Regev from the Broad, and Tommy Kaplan from the Hebrew University. And what we did in this work is, as I told you, we compared dozens of mice of both wild type and 5XFAD, and we profiled over 200,000 cells using single nuclear RNA sequencing. And the first thing we did is actually to build the cellular map of the hippocampus in both wild type and Alzheimer's disease, and just ask in the most unbiased way, which cell types do we see that are dramatically changing? So I'll jump directly to the results. Here you can see the output of our algorithm, where on the left, you can see the map of the hippocampus of the wild type mice. And on the right, we can see the Alzheimer's disease. So to explain these graphs, every dot is a single cell. And this is a two-dimensional embedding of our very complex RNA measurements. And every cell, every two dots are close together if we have two cells that are similar in the sense that they have similar RNA molecules. And these cells are also color-coded based on a clustering algorithm. And we can see that every cluster, every color here captured a really unique population of cells. So we found multiple different types of excitatory and inhibitory neurons. And we also found all the different glia cells that we expect to find in the brain, microglia, oligodendrocytes, astrocytes, different endothelial cells, and so on. So the first thing you can see is that the wild-type brain and the Alzheimer brain are actually, at a first glance, are very, very similar. So this was actually surprising to us, since we're profiling here mouse that are have quite advanced Alzheimer's disease. They're 7 months old, which means in this mouse model, they already show sign of cognitive decline. So one thing we already saw is that microglia cells show a dramatic increase in their numbers and their frequency in the Alzheimer's brain, and also a switch to a new disease state, which matches the previous results I actually discussed before. What we've also noticed is that astrocyte cells, marked here in the black lines, actually show the very dramatic change in their number and frequency. So we really were fascinated by these changes. So let me just say a word about astrocyte cells and why we were actually excited that we found such dramatic change in them. So astrocyte cells are star-shaped glia cells that are the largest cellular component in the central nerve system. And they have so many important roles in maintaining brain homeostasis that any change in astrocyte cells could actually be very detrimental for the brain's function. So they have multiple roles in maintaining ion homeostasis. They're known to be supporting neurons, both the neuronal function and metabolically. They're critical for the integrity of the blood-brain barrier. They've been extensively characterized in the sense of their role in brain repair, and they have many, many different roles in both health and disease that's already been reported. So in order to really understand what our finding, we took these astrocyte cells and we focused on our analysis on them. So this is actually analysis of the astrocyte cells only. And what we see here is, again, a two-dimensional embedding of, this time, only the astrocyte cells, but using a different algorithm that enables us to look not at discrete changes, but rather continuous changes along a population of cells. So again, on the left, we see the map of the astrocytes in the wild-type brain. On the right, we see the map of the astrocytes in the Alzheimer's brain. And every dot here is a single astrocyte cell. So what we clearly see is that in wild-type brains, the astrocytes range between a homeostatic state to a state that is GFAP high, which is known to be a reactive astrocyte marker. So they seem to be ranging between they seem to be ranging between a state, a homeostatic state, to a reactive-like state, which this is what happens in a wild-type brain. What do we see in Alzheimer's? In Alzheimer's, we see the same range between a homeostatic to a reactive-like state. But we also see a completely new state that emerges that is completely unique to the disease state. You can also see it here quantified in these bar plots. So what we found is a disease-associated state, which we called DAA, disease-associated astrocytes, that are unique to the Alzheimer's brain, and we cannot find them in the wild-type brain. So we're very intrigued by who are these disease-associated astrocytes. So since we have actually thousands of RNA profiles associated with these cells, we could do a very detailed pathway analysis and show that generally the reactive-like states, both the disease and the non-disease, are both associated with an increase of inflammatory immune-related genes. But specifically in the disease-associated state, we see a very strong connection also to reactive oxygen species, aging-related pathways, and the complement cascade. Interestingly, we also see an upregulation of metabolic genes related to lipids biosynthesis. When we then use markers for this new disease-associated astrocyte state in order to look at the position of these disease-associated astrocytes within the brain, we confirmed on the protein level that indeed we find disease-associated astrocytes only in Alzheimer's brains and not in wild-type brains. And when we look at the positions of the disease-associated astrocytes, we see them very closely positioned next to A beta plaques, which are marked here in gray. The astrocytes are marked in green and red. So overall, what we found is we found specific pathways related to stress response and inflammation in the disease-associated astrocytes, and we also find them in proximity to A beta plaques. So both of this is very intriguing and kind of suggests many hypotheses regarding their potential role in Alzheimer's disease. But in order to better understand this, we actually wanted to see how early do these disease-associated astrocytes appear during disease progression, and what happens as the disease continues and to progress. So we extended our analysis to look at a very longer time course, ranging from 1.5 months old mice, which are very young, before any signs of pathology or cognitive decline, and going all the way to 20-month-old, which are already aging mice. And we performed the same analysis using single nuclear RNA sequencing, and then we took the astrocyte cells and looked at them separately using our clustering analysis. So here you can see a similar two-dimensional embedding of the astrocytes, but every graph here is a different mouse model at a different age. So the top row is all the wild-type mice, and the bottom row are only astrocyte Alzheimer mice. And we can see that overall in the wild-type mice, we don't see a dramatic change in the landscape of Alzheimer's, of the astrocyte cells. But what do we see in Alzheimer's disease? We clearly see the emergence of the disease-associated astrocytes, and we can actually quantify the frequency of disease-associated astrocytes out of the entire community of astrocytes, and we see a constant increase in the disease-associated astrocyte proportion as the disease progresses. Interestingly, we find these cells as early as 4 months old mice, which is before any signs of cognitive decline in these mice, suggesting that they actually appear early during disease. On top of that, we also see a small population, quite rare, but still existing, of these disease-associated astrocytes in aging wild-type mice, suggesting that we might have here an interplay of both genetic factors and aging-related factors that could lead to the appearance of disease-associated astrocytes. What else is that if we look at other population of astrocytes, specifically the homeostatic population of cells, we see a constant decrease in the homeostatic population of astrocytes as disease progresses. So this actually very intrigued us, and it suggested to us a model in which the disease-associated astrocytes appear from a switch in the homeostatic astrocyte cells. So overall, we don't see an increase in astrocyte cells, rather we see a switch from a homeostatic state to a disease state. So this switch was actually a little surprising because if we look at the molecular profile of the disease-associated astrocytes, they're highly similar to these reactive-like physiological cells. So to prove that indeed the disease-associated astrocytes stem from a switch from the homeostatic cell, we performed we developed a new algorithm to perform a data simulation where we predict for every astrocyte cell it's a unique origin cell from a previous time point, assuming there is a switch from one cell to the other. And we can then generate these graphs showing which, for every cell cluster, which cellular population most of these cells are coming from, showing that indeed most of the disease-associated astrocytes emerge from the homeostatic cells and not from the reactive-like cells in the previous time point. So overall, what I've showed you is that using single nuclear RNA sequencing and generating maps of the Alzheimer's brain, we were able to find a unique population of astrocyte cells that is switching in disease from a homeostatic state. These disease-associated astrocytes are positioned next to A beta plaques, are associated with many pathological pathways, and most excitingly, we see that they actually appear early during disease progression and keep increasing as the disease progresses, and even appear with natural aging. So overall, this evidence suggests that astrocytes might also play a critical role in Alzheimer's disease progression, but it also leaves many open questions relating the functional role of astrocytes, their interaction with other cell types such as microglia cells, oligodendrocytes, and neurons themselves, and also what is the relevance of our finding in mouse models to human brains. So I can't answer all of these questions right now. A lot of them are actually work in progress in my lab, but I'll just show you a little bit of our new findings in human brains. So this is actually a new collaboration that we have with Professor Phil de Jager from Columbia University, with Mariko Tago and Vilas Menon, Aviv Reged from the Broad Institute, David Bennett from Rush University, Esti Agerlotem from Ben Gurion, and which is all led by my student, Anael Kain. So what we did together is we used a similar approach of single nuclear RNA sequencing to profile the healthy brain and the Alzheimer's disease brain, this time of human post-mortem brains of aging individuals. And we generated these very highly detailed maps across 24 individuals. So the individual range between healthy and Alzheimer's disease, both men and women, all are aging with different degrees of pathologies. And these maps are actually amazing because they're so detailed. We could really drill into and understand the cellular diversity, the diversity across individuals, diversity related to sex and age, and also, of course, to disease. And here, I forgot to mention that we are actually profiling the dorsolateral prefrontal cortex of these individuals. And as I actually showed before, this is a very similar map to what I showed before, where every dot is a single cell, and they are color-coded based on the different clusters that each capture a different population of cells in the brain, many different types of neurons, and also all the different types of glia cells, astrocytes, microglia, oligodendrocytes, and also endothelial cells and others. So what we can see now is we can actually try and start and look at the link between all the different cell types and their link to Alzheimer's disease. And what I'll do now is I'll only show you our results on the astrocyte cells to continue our story. So when we zoom into the astrocyte cells and actually perform an individual analysis of them, you can see them here actually generating this very nice map of astrocyte cells, and they're clearly dividing to very distinct states, even though we can already see even here that we see a continuous transition across cell state, that it's not binary, very similar to what we've seen in the mouse. So here again, we can actually identify a more homeostatic state and compare to different reactive-like states. So because this is actually human brains, there's much more diversity in both genetic and in lifestyle. So we're expecting to see much more diversity, which is indeed what we're seeing here in these astrocyte cells. But we could already look at these reactive states and showing that all the reactive states compared to the homeostatic are already upregulating immune-related signaling, similar to what we've seen in the mouse. But we were very intrigued by these fibrous-like reactive astrocytes that are not only expressing immune signaling genes, but also a lot of oxidative stress, metabolic signaling, and also very interesting senescence pathways. So we wanted to see if there's any link between Alzheimer's disease and the different states of astrocyte cells. So just to remind you, in the mouse, we found a disease-associated astrocyte state, yet we also saw signs of it as might be dependent also on aging itself. So we weren't expecting to find a state that will be unique only to Alzheimer's disease when we're looking at the human brain. So let's see what we found when we split the cells to healthy individuals on the left and Alzheimer's disease on the right. And so orange cells would be the cells associated with either Alzheimer's or healthy. We can clearly see a very dramatic increase in the number of the fibrous-like reactive astrocytes. So because we only have 24 individuals, our statistical power is relatively limited. But what we next did is we actually wanted to see how do all of these cellular states divide across individuals. And to our surprise, what we found is that they're really not dividing uniformly across individuals, but rather most individuals have either a very highly enriched brain with only homeostatic astrocytes, or they have mainly different sets of reactive-like astrocytes. So again, similar to what we saw in the mouse, we see a dramatic switch between a homeostatic state to a reactive-like state. So in order to increase our statistical power beyond the 24 individuals, we actually applied a new computational approach which enabled us to take the single cell profiles from 24 individuals, where we can really infer the different cell types, different cell states, and their proportions across individuals, and now take bulk RNA sequencing from 640 individuals, a much larger cohort, and use a deconvolution approach to infer the cellular proportion, not only of different cell types, but really of the different cell states. So we can really say what is the proportion of the different states of astrocytes in each of these 640 individuals. So this analysis actually helped us confirm both that we indeed see a very dramatic switch between a homeostatic state to a reactive-like state in human brains, but also that we see a statistically significant increase in the fibrous-like reactive astrocytes in individuals that are cognitively declined and have Alzheimer's disease. So in fact, this story actually goes well beyond astrocytes. And when we repeat this analysis across all the different cell types, we see interesting switches in cell states across multiple individuals, which we can connect to Alzheimer's disease, including a very significant contribution to oligodendrocyte cells that we are now finding and linking to Alzheimer's disease. So to summarize, I showed you today how we're using a single nuclear RNA sequencing approach to build very detailed cellular map of the Alzheimer's brain in both mouse and humans. And by using these models, we can identify disease-associated astrocytes that we show in mice appears very early before any signs of cognitive decline and increases as disease progresses, and that we also find a similar yet different population of disease-associated astrocytes in human brains, and also that we find coordinated changes in cell states well beyond astrocytes across multiple different cell types, which is leading us now to much better understand not only a single cell type, but rather the entire cellular environment of Alzheimer's brain. So I want to thank you for listening, and I think I thanked most of my collaborators along the way. I also want to thank again my amazing lab, which is really great to work with and a lot of fun, and to thank the brain donors from the RoseMap cohort at Rush University, which are making such an important contribution to science. So of course, thanking also my um, funding sources, and thank you for listening.